Good morning, everyone. Thank you for coming out, to those of you who are in the room in this beautiful, it's kind of like Ireland, I was saying. It's very, very apropos of that. I'm Melissa Geller. I'm a GYN oncologist here at the University of Minnesota, and we're hosting Dr. Eimear Dolan here today. So I had the great opportunity to spend about 6 months in Dr. Dolan's lab when I was on my Fulbright scholarship in Galway last year. Dr. Dolan is in the biomedical engineering group at the University of Galway and has really been there since 2019. She's a Royal Society Research Ireland fellow and a Research Ireland starting laureate. And to date, she's been involved in successful generation of over $53 million in research funding. She publishes frequently in top-tier journals, including Science Robotics and Nature Communications, as well as Advanced Science and Healthcare, Advanced Healthcare Materials. These are journals that I often have not read in the past, but now read more frequently. She's co-founder of University of Galway's startup company called Veda Medical, and is really building on 2 patent applications currently. Dr. Dolan's previous successes have really been due to engagement with international clinical experts, as well as academics and medical device industries. She's really passionate and motivated to design solutions primarily for women's health, as well as for diabetes. And her group is focused on really the fundamental and translational research of innovative medical implants. So they're currently designing medical implants to address long-term therapeutic needs for both ovarian cancer, as well as diabetes and cardiovascular diseases. She works really, what I found, at the interface of both engineering and medicine. And I think this relationship really exemplifies, how critical such relationships can be for people in medicine like us, and to overcome some of our everyday significant barriers that we encounter. So I'm thrilled to have her from Galway visiting us, and I hope you enjoy this talk.
Thanks so much for having me, and I'm delighted to be here. So I'm going to start with a little bit of geography. Just figure this out here. So Galway is a small little city on the west coast of Ireland. I like this picture here, which is a picture of our university when it was established in 1850, about 1849. And I think it's kind of cool that you can see like maybe 4 or 5 little houses in the background here, and it's all green fields. And this is our university now. So the university has grown so much over the last number of years, but as has the city around it. So Galway is internationally recognized as a medtech hub alongside you guys here. We have 8 of the world's top 10 medtech companies that have a physical presence in Galway, and we produce 80% of the world's stents. So we have a few things, and Dr. Geller mentioned that we work or try to work at the interface of engineering and medicine, and we are also kind of tightly aligned with local medical device industry. And I think we have that ethos in Galway because of kind of a few flagship programs. So our program in biomedical engineering in Galway was the first program in Ireland. It kind of grew alongside the medical device industry. And we also have a program called BioInnovate, which is an innovation program for medtech. And it's co-led by the School of Medicine and the School of Engineering, where we send fellows into the clinic for 3 months, and they observe challenges and then come back to us with a list of needs that they try to then develop into an innovative solution. And this has really been a really successful program for us, and has led to a lot of startup companies locally. So I'm a biomedical engineer. I started my educator training in Galway with a BE in biomedical engineering, and I stayed for a PhD. I did my PhD with Stryker, who are another multinational company, and I had great experience of managing a negotiation of doing academic research, which is aligned with industry needs and timelines. They're a great company to work with, and they really valued the importance of fundamental research to then be included in their learnings for the development of the technology. So I think that was really great training for me. I was lucky that I had a travel fellowship as part of my PhD, which allowed me to spend 6 months at Notre Dame and 6 months at NYU. So following this, I did some postdoctoral training for about 4 years, maybe, in Trinity College Dublin, the Royal College of Surgeons, Ireland, and MIT. The Royal College of Surgeons is a medical school, so I was one of not that many engineers that were based there. So I had a great experience working alongside clinicians there and kind of incorporating the voice and the importance of the clinical experience in our designs. And then I started my group in Galway in around 2018 and 2019. I now lead a research group of 12 researchers, and we are interested in both, fundamental discovery as well as translational and applied research. So we have about four researchers in my group who are purely working on commercial projects, with the goal of future spin-out. And as Melissa mentioned, I'm involved in another spin-out company called Fada Medical, which I'll talk a little bit about in a few minutes. So our mission really is to design innovative medical implants for long-term therapeutic needs and coupling medical implants with novel therapies. And I think that second point there is really important to me, and I think it's very relevant for the work I'm going to talk about today around delivery of therapies for ovarian cancer. We're not tied to a specific disease state. We have projects in cardiovascular disease, diabetes, ovarian cancer. I have a new student starting on colorectal cancer this autumn, and we just started another project on bone, so we're kind of spread widely, but I think we learn a lot, so particularly from the area of therapeutic implants for diabetes. A lot of the learnings that we had from that area we are now applying to therapy delivery for ovarian cancer. Of course, very different diseases and different therapeutic needs, but a lot of similar challenges and solutions that we have developed along the way. So ovarian cancer, as many of you are aware, it's a big problem. It's the 8th most commonly occurring cancer in women and one of the most lethal. Gold standard treatment is surgery and IV chemotherapy. And this is a very old graph from 1995, but I think it's nice to show because it's important to show, I guess, because the overall survival hasn't improved too much since then. In the US, I think you're getting close to around 50% 5-year survival, but across the world, it's probably somewhere between 30 and 50%. When I started working in this area first, I was I became very interested in IP, or local delivery of therapies for ovarian cancer. It was interesting that there was 3 pivotal trials in the 90s and the 2000s, which showed that IP delivery of chemo improved overall survival in all 3 studies. And it led to the NCI releasing this alert to recommend IP delivery become the routine delivery route. Now, this hasn't been adopted, and it's not the preferred delivery route, and it's only really done in certain centers of excellence. And the reason for this, I think, is that this study explains the problem very clearly, in that only 40% of patients completed all 6 rounds of chemo, of IP chemo, and the major problem or reason for these people being pulled off the study was there was catheter related issues. So people are not continuing their life-saving treatment because the catheter fails. Now, that was a long time ago, I guess, but more recently there has become growing evidence to support IP delivery of therapies for peritoneal cancers again. So this 2023 trial compared IP delivery of carboplatin, and they showed an overall survival that IP was better. And then that led to this review article that came out earlier this year to say that IP chemo is back. And in the field of ovarian immunotherapies, these are recent papers that came out too. There hasn't been a major breakthrough in ovarian cancer yet, but this paper is very recent on NK cell therapy for colorectal, and they really talk about the benefits and the importance of local IP delivery. So I guess this is the clinical trial that was published, and then this is a perspective piece that was written about it. Importantly, on ovarian cancer, this is probably one of the most promising trials that has come out for ovarian cancer on this IL-12 gene therapy, and it showed that a 13-month survival benefit, and this therapy was delivered IP, and they've now moved forward to a phase 3 clinical trial. So there's kind of more and more evidence building on the importance of IP delivery of different therapies to improve outcomes. Now, interestingly, when we look at the catheters that have been used to deliver these types of therapies, some very novel therapies that have taken millions of dollars to produce, there hasn't been much innovation on the catheter side. Now, the rates have improved. The trials I mentioned earlier showed 42%, only 42% of patients completing the therapy. It has improved since then, but the devices are kind of the same. So I think it's maybe people have, these centers of excellence are getting better at using them, but I think that there's been, we can do better on the catheter side. I think they're just using the catheters that have already been established. And interestingly, all 3 of these catheters were not designed originally for therapy delivery to the IP space. And I have a nice quote here from my friend and colleague or collaborator, Melissa Geller, really saying, stressing the importance of the design of new delivery devices for these novel therapies. So when I started working in this area in 2022, we wrote this review article, and we really set a goal of designing a replenishable implant for long-term, minimally invasive access to the peritoneum. Now, unfortunately, I can't show a picture of our device or implant right now because we're finalizing a patent application on it that I had hoped to get finalized before this, but it hasn't come together. But I guess our device was designed to overcome the limitations of the catheter-based systems that I mentioned earlier, from mechanical mismatch to the surrounding tissue, limited porosity, non-uniform therapy distribution within the peritoneal cavity, and minimize the complications that we've seen with these previous catheters that have been developed. So our design is a porous reservoir that's placed in the peritoneal cavity, connected to a catheter which tunnels to a subcutaneous port. And in our mouse model, we have the port located at the back of the neck of the mice, and we found that was the ideal location to prevent the animals from interfering with the port. So a student of mine, Aveen Sheedy, got a Fulbright scholarship in 2023, and she spent a year over here working with Dr. Geller and Dr. Felices and Dr. Jeff Miller, and she tested the function of these devices in the ovarian cancer mouse models that you guys have so much experience with over here. So our first study was basically to determine if this implant could in fact deliver anything to the peritoneal space. So we had a lot of experience designing implants for the diabetes field, and in that case we were always implanting these devices in the subcutaneous space, so we had never gone into the peritoneum. So in this case we implanted our device, and almost 2 weeks later we delivered ovarian cancer cells by IP injection, and then we started delivering Genhance, which is just a fluorescent probe. We weren't looking for a therapeutic effect in the study, we just wanted to visualize if we could get something out of our device into the surrounding space. So we can see here in blue is the IVA's imaging of the cancer growth over time. So we're showing that the ovarian cancer is establishing over time, and then the red is delivery of GenHance, which is our fluorescent probe, and we can see that we're getting consistent delivery over time in this mouse model. And what's interesting is we can really see, even at day 21, that we're getting very good dispersion of the therapy throughout the peritoneal space. So it's not kind of localized. We're really getting a kind of a washing, which I think is important, and it was one of the features that we wanted when we designed this type of implant. Next, we wanted to see if we could deliver cell therapies through the implant. And in this case, we compared NK cell therapy, an NK cell therapy regimen delivered via our implant or via IP injection. And we compared therapy delivery to control delivery of just saline. So you can see here with our IP injection, we're getting an increase in tumor burden over time in blue in our both the control groups, and then our therapy controlled the tumor somewhat when delivered by either injection route. And when we directly compared IP injection to implant delivery, we saw comparable control in the earlier time points, but in the later time points, we found a significant improvement in control of tumor burden when the therapy was delivered via our implant compared to IP injection. When we designed the study, we kind of wanted to be as good as IP injection, which is kind of gold standard for mouse models. And we were delighted that we saw an improvement, and we think it's because of this, dispersion of therapy throughout the cavity, which we are very excited about. So next, we wanted to see if we could use our implant to understand the peritoneal environment. So ascites is the buildup of fluid in the peritoneum, and many women with late-stage ovarian cancer develop ascites, as well as other peritoneal cancers also have ascites as a major side effect. But we were interested to see if we could learn something from this ascites fluid. So if we have our implant long-term implanted in the peritoneal cavity, can we use it to sample this peritoneal fluid and try and understand disease progression and potentially therapeutic effect? Now, some of the clinical trials are doing this with the IP catheters that are currently being used, but it's not being done in mouse models because there's no devices, there's no catheters that have been developed for mouse studies. It hasn't been scaled down. So we think that we could really learn a lot from the ultimate aim is to use it for clinical studies, but I do think there's definite benefit for using this type of implant to understand our mouse models too, and understand and refine our therapies as they're being developed. So this is really nice data from our collaborators here, where you guys have shown that when you isolate NK cells from the ascites and compare it to NK cells isolated from the blood, there are differences. They're phenotypically different and they're functionally different. So I guess it argues the point of, we need to really understand what's happening in the peritoneal cavity to understand how our therapies are actually having an effect, or to understand our disease progression, that blood-based markers, are not enough, or not good enough, or not accurate enough in this case. So this was our kind of first very early proof of concept study to investigate if we could sample. So we did a very similar timeline as I showed previously. In this case, we delivered less NK cells, but the important part is that we sampled peritoneal fluid through our implant on day 7. So it's kind of like a peritoneal lavage, which is only possible in current animal studies at the end point. So now we were doing it on day 7. And we compared again our IVUS imaging, or BLI of our treatment of our ovarian cancer growth, and we showed that we got a significant improvement with our treatment group compared to our control. We isolated NK cells from the peritoneal fluid that we sampled at day 7 and ovarian cancer cells. So the ovarian cancer cells, had a similar trend as we saw with our IVUS imaging, and the ENK cells. So we only delivered ENK cells in our therapeutic group, not in our saline control group. So we had no ENK cells in our saline control group, but we could isolate them, we could identify them, and we also looked at the functionality of these NK cells when isolated on day 7 compared to day 27. So our endpoint either isolated through our implant or via the traditional method of IP lavage, and we saw no differences. So we're excited about this, and we think that we can really use this implant. In this study, we just did one sampling time point at day 7, but ultimately we want to do it potentially weekly, so we can track what cells are in the peritoneal fluid, how are they responding, and maybe ultimately we could use it as a guide for a real-time maybe adjustment of the therapeutic regime to really control ovarian cancer. Another thing that we're interested in is the physical forces that happen as a result of the buildup of ascites. So, there's been reports that you can get a buildup of pressure of up to 25 millimeters of mercury as a result of the buildup of ascites. Some patients get up to 5 liters of ascites fluid drained, and they might do this pretty regularly. And there's huge strain on the anterior abdominal wall, as well as altered interperitoneal fluid flow. So the pathological environment as a result of the buildup of ascites, in my opinion, has to have an effect on both the immune cells in that environment, but also the cancer cells. This is a really nice study, I think, by our collaborator, Professor Sharon Stack at Notre Dame. And it's kind of one of the only studies that I've come across that has actually really asked the question about the physical environment as a result of ascites. So they this is a simple but very nice study where they injected ovarian cancer cells in either 1 ml or 5 ml of fluid. So AA here means artificial ascites, and they looked at adherence of the ovarian cancer cells to the anterior abdominal wall, the mesothelin, and they showed that when they had their artificial ascites model, that they got much more ovarian cancer cells adhering to the wall. So this was a very short term. So the mice actually ended up clearing the fluid pretty quickly, I think between 24 or 48 hours. But even during that time, the strain of the wall caused structural changes in the collagen, which was more attractive for the ovarian cancer cells to adhere to. We've started looking at, so that's just looking at strain of the wall, but as a result of pressure too, it's very difficult to decouple these physical forces. But we've started looking at pressure, and can we use our implant to understand pathological peritoneal environment? So if we could use our implant to measure increases in pressure, and maybe we could start to identify, okay, this is the threshold. If we let the pressure rise in the peritoneal cavity above this threshold, the cells become more chemo-resistant, metastatic, I think this question of pressure has not really been asked, or I haven't come across it being asked in the literature. So this is just a very preliminary data where we use our implant to measure increases in pressure. We place the implant in different depths of water, and then we use it to measure pressure. And we've started culturing our cells just in these slides where we increase the pressure and looked at things like number of live cells, metabolic activity. This graph came to me from my postdoc late yesterday evening, so it's very fresh data, and we just use the suspension cell line. He just used the suspension cell line that we had growing in the lab at the time. But we are starting to do this now with ovarian cancer cells, but I also think it would be valuable and interesting to do it with immune cells too, to look at their functionality. So we just submitted a DOD grant with Melissa Martin and Sharon Stack to really start asking these questions or around understanding these physical forces and the physical environment in ascites. So we have done some work on scaling up our implants in the past. So when I started working on these devices a number of years ago, I had seen a lot of stuff being developed in the lab that potentially worked in small scale batches, but when it came to scaling up like biomaterials or devices, they became a challenge. So we really didn't want to design something that could only be used for, we'd have to go all the way back to the start again if we wanted to change a feature. So we came up with a protocol where we could very easily make these devices, add or remove features very quickly, and we could quickly scale them up from small animals, scale them up from small animals to large animals and ultimately to humans. We published this paper in 2020 where we just showed 3 different versions of a device. So a cardiac, interperitoneal and intramuscular device. They all had the same features. We wanted them all to be minimally minimally invasively delivered. We wanted them all to have a therapy reservoir, and in this case, we had a bioadhesive reservoir. So the idea was that we deliver them minimally invasively, and then we'd adhere them to the location that we wanted to adhere them to, minimally invasively. And you know, we this last one, I don't know, I mentioned in my talk earlier that I don't know why we'd ever need a device in that shape, but we just put it in that shape to show that we could do multiple shapes. But if we look at this is an example of the peritoneal implant. The yellow here is the bioadhesive, the blue is a therapeutic reservoir. We wanted to make the devices from a single material because we wanted it to be easily translated in the future, and it would just make our lives easier if it was made from a single material. And we picked thermoplastic polyurethane because we can laser cut pores into this material. So we could make the device and then decide where we wanted it to be porous, and we could very easily change that porous region. And then this video here is just of the implant being delivered after delivery, minimally invasively, in an acute pig study, and we could visualize the device after delivery with this contrast agent, which we have delivered. Now, we are a little bit obsessed in my group with the foreign body response, and I think we need to be as medical implant designers. So unfortunately, the body does not recognize the difference between a therapeutic implant and something that is dangerous, like a splinter or a bullet. So we really need to understand the foreign body response, and we need to really, I think it's one of the most interesting things about biomedical engineering as opposed to other engineering disciplines is that everything we do, and if we implant it, we need to understand how the body's going to react. So we know what we've done so far on the ovarian cancer side has been using NSG mice, so immune compromised mice. And the reason that we've started with that is because we're using human ovarian cancer cell lines and human NK cells to deliver and investigate the therapeutic effect. But for medical device designers, we need to understand how these implants will perform in an immune competent setting. Now, we know, we've really done a lot of work on understanding the foreign body response over the last number of years. And we know that neutrophils are the key driver in this response. And, women who are receiving chemo often do have low neutrophil counts. So potentially, they might not have a robust foreign body response to these medical implants. But nonetheless, we do need to understand how these devices or implants will function in an immune competent setting. So we have a lot of experience, as I mentioned earlier, designing implants for in the area of diabetes. And we use this C57 Black 6 mouse, which is a mouse model that is accepted in our field as it can truly recapitulate the foreign body response in humans and non-human primates. So we established this model in our group a number of years ago, and we have this protocol where we deliver insulin through our implants, and then we measure changes in blood glucose levels. So if the insulin can come out of the implant, then it will have an effect. So you can see here in black, baseline, very nice response. So the insulin comes out, it has a nice change in blood glucose levels, and then over time this starts to dampen. And I think this graph, when we saw it, we were delighted, even though, we are showing failure of our implant over time, but it's often really challenging to design an animal model that captures failure. So, we know these implants fail in the clinic, but, they might perform perfectly in our animal model. And when I started my group, first we were trying to establish an ovarian cancer mouse model, and we just couldn't get the cancer to grow, and my students were like, it's great, we've cured cancer, but we really need to establish these models that are functional and work. So this graph I think is really important to me because it does show failure over time, but even better is that we designed a solution to overcome it. So we have this device that we call it a mechanotherapeutic platform, so mechanical therapy. And basically what it does is it pulses, it actuates. Actuation is a real engineering term, but it has two kind of mechanisms of action. So this top one is that we actuate it, or we activate it for 10 minutes a day, every day that the device is in use. And we've shown that this daily actuation prevents the buildup of the fibrous capsule around the implant and allows the implant to function long term. And then we have this other mechanism where in this scenario, we imagine that we implant the device, let a fibrous capsule form around it, and then activate it to just push or remove any blockage that's in place. So we've looked at both of these mechanisms. So this is the second burst activation, where we have 2 devices here in the back of a mouse. And again, we're using Genehance, the therapeutic the fluorescent probe, not therapeutic, that I mentioned earlier. And we've shown that in our control group, after a certain amount of days, 24 days here in a rat model, we're not getting much therapy coming out of our reservoir. Whereas if we activate it, or we burst this device out, or this therapy out, and push it into the surrounding tissue. Oh, sorry, that's just that video playing again. Sorry. And then this is our daily actuation. And here at 8 weeks, we have our red here in control, where we're shown the insulin's not coming out of the device. It's not having an effect in the surrounding, it's not getting into the bloodstream to change blood glucose levels. Whereas in our 8-week actuation group here, we're getting a really nice response. We also wondered if we could just activate or actuate our device for 3 weeks. So if we could just focus on the acute phase, would that be enough to have a long-term benefit? And it did have some benefit, but it wasn't as good as daily actuation, which really tells us that we need to be actuating this device daily while it's in use to maintain its functionality. And excitingly here, we showed that there was no significant difference in the function of the device after 8 weeks implantation, which is shown here, compared to a device that is implanted for 3 days. And this 3-day time point is really important for diabetes, because that's the time point that a lot of these medical devices for diabetes fail. So we have started a campus-based spin-out company called Fada Medical. So Fada means long, Oscailgar in Irish. So the idea is that we're extending the life of these types of implants. So this is an infusion set cannula, which is the part of an insulin pump that is transcutaneous, and these are changed every 3 days. I mean, there are new devices which can get to 7 or potentially 10 days, but the majority of them are changed for 3 days. So our vision is like, what if we could get to a month? What if we could just let these devices, leave the devices in for a month? It's 12 changes a year, which is important. And I guess for us, we you might think, okay, the infusion set, people just get used to it, but we're really interested, or I'm particularly interested in, it's challenging to deliver these types of infusions at cannulas in children. And if we could, even change, double or reduce by half the amount of time they have to do it, it would have a real impact on people's lives. So I guess that's what we're trying to do with Fada Medical, building on patent applications that I'm a co-inventor on, and we're now actively raising a seed round to take this to the next level. So I guess what we've learned a lot from as I mentioned earlier, from the field of diabetes, that we're now applying to ovarian cancer. So we're looking at this mechanotherapeutic platform for ovarian cancer, and we're establishing this mouse model with ID8 ovarian cancer, so syngeneic cancer cells in our group in Galway. I will mention, actually, I have this C57 black 6 mice here, which we have experience with in the past, but we've just come across an albino version of those mice that the black fur does interfere with. Just in case anybody is interested in using those mice, the black fur does interfere with the IVUS readings over time. So the albino ones, we are hoping will solve that problem for us. But we're establishing that model in our group at the moment. So I guess a question one might ask is, but can we use this mechanotherapeutic platform to deliver cells? And will this actuation, damage the cells as we're delivering them? The answer is we're figuring it out, and we think so. Like the worst case scenario is that we would actually pulse actuate the cells out of the device. But in reality, what we might do is actuate the device to kind of remove any blockages. And then, once we're comfortable that the blockages have been removed, then we deliver the cells. So in this case, we've kind of looked at the worst case scenario if we are actuating the cells out, and we're looking at one of the real benefits, I think, of these types of devices that I'm talking about is that we have a reservoir in the peritoneal space which is connected to a transcutaneous port, and we can access the port to change our actuation regime. So the two mechanisms that I talked about earlier, burst actuation versus daily actuation, it's the same device. We're just plugging it into a different program on the outside. So we can change, we can adapt it. We can change our actuation profile depending on which stage of the temporal foreign body response we're dealing with. And I think that's the key difference to some of the other solutions that are being developed at the moment to tackle fibrotic encapsulation of medical devices. They can only target one specific stage of the fibrous capsule. So maybe anti-inflammatories or something that is delivered earlier on, but then when they're released, then what happens after that? We've shown that when we stop actuation, we still get the formation of the capsule. So it doesn't get over by doing something at the earlier stage. It doesn't mean that the device will not be encapsulated later. So I guess that's what we're doing here as well. We're looking at different actuation profiles that might benefit or might ensure that we're keeping the cells that were being delivered alive. So here we're just looking at primary mouse NK cells delivered by a triangular platform and a square platform. And basically, I'm just showing here that we can change the actuation profile in order to maximize the viability and the functionality of the delivered cells. So I think that is my last slide, and I guess I hope I've given you a good overview of the work that we're doing and the overall mission of my group, which is to design innovative implants for long-term therapeutic needs and really coupling those implants with novel therapies, and the importance of kind of not forgetting about the device or the way it's going to be delivered until the very end, because I think it does need to be involved the whole way along in order to really design the device to maximize the therapeutic effect. And this is just my research group and my collaborators and my funding, and thank you so much for listening.
I've got microphones to pass around for anyone with a question. Just raise your hand.
Thank you. That was really interesting. I wonder, do you know if you could engineer an enzymatic system to break down the fibrous capsule as it's being made? So whether you could infuse something like collagenase or something like that, or even if you're using cells, actually have the cells make their own enzymatic activities to break down that barrier. Do you think that's feasible, or do you think that it would create all sorts of havoc by breaking down connective tissue in the peritoneal cavity?
Yeah, I have come across people who have looked at that, and I definitely came across a clinical trial that was doing something like that a couple of years ago. I think it is a good approach. I guess for us, we wanted to try and minimize the amount of drugs that we're kind of putting in there, and we're trying to minimize the inflammation. So it is one approach that has been looked at, and I think you do need to be delivering it long term, I think, and I think then you need to be investigating the effect of that agent with your therapeutic, which needs to be tested then very thoroughly before you're doing it. It is definitely an approach that has been looked at. Now, I've never seen it. It hasn't gained much traction for whatever reason. It does seem like a logical way of doing it for sure. And I have heard people kind of talk about it intermittently, but it's never really gained that much traction for whatever reason. So I kind of think that maybe it's the interference with the therapy that's being delivered. But that's just my opinion on it.
Okay, we've got a question from our virtual audience. For the different actuator waveform, is there any indication that rate versus duration are more dominant factors for cell delivery and viability?
Rate versus duration. Yeah, rate, like, so I think trying to minimize the like trauma, I guess, on the cells as they're being delivered is the most important thing. So we found that kind of slower delivery is better. And, you know, there's no reason really for us to actuate very quickly to just push the cells out. You know, maybe there is a benefit longer term to have something very slow release over time. And we haven't looked at that. We're looking at, you know, okay, how do we get them out into the peritoneal cavity quickly, while minimizing the trauma onto the surrounding cells? I hope that
All
answers
right, that question was from Paulo Provenzano. Here's one question at the back.
Hi, thank you for the great talk.
Thank you.
A question about metastasis from the catheters.
Mhm.
You guys are simulating agitating the device to get rid of the fibrous capsule. Are you also then hypothesizing that that could reduce the metastasis spread that comes through the catheter?
Through the peritoneal wall.
Correct.
Is that, yeah. So I think our ultimate aim is to have an implant that we put in and leave it in long term. So I think some of the challenges with the current devices is that they're being put in and, you know, they're going through the initial stage of therapy, but then we need to pull them out. So we want them too anchored, and because we do need to take them out again. Our ultimate vision is that we could put a device in at the initial surgery, we use it for the initial treatment, and then we leave it in for recurrence. So then, you know, we really want it anchored in there. We have not seen any tumor tracking through the peritoneal wall, but I know it is a challenge. And I think it's because the devices are, you know, the catheter tubing is kind of moving, it's not fully anchored in there. But I think that if we really anchor it to the peritoneal wall and have it sealed, I hope that that can really minimize that. I know Melissa and I talked about this a lot, and I think that the ascites probably does have an effect there too. So, you know, as the pressure builds up, maybe that's one of the reasons why you do get tumor tracking along that track. But we don't know, and we are going to be looking very closely at it in our animal models.
Thanks so much for that talk, and it's great to see the translation of the device.
Mm-hmm.
And so, as a follow-up to kind of this goal of kind of long-term implant, just to leave it in there, do you have to, with this particular device, do you have to run into any particular concerns that are different from other devices, such as biofouling for kind of long-term use? And thinking about now, particularly in Europe, some of the regulations on some of these like fluorinated chemicals that have to go into these devices, which are sometimes used. Is that affecting how your company assembles these devices?
We are, like, I guess the manufacturer is something that we have not started yet. At the moment, we're for the company that Father Medical, is that kind of what you're asking? Yeah, so we're still campus-based. We're making the devices in the lab, and we're testing them in large animals at the moment. But it is something that we really need to, when we scale up and you know, manufacture for clinical use. Yeah, but there are, you know, there's established protocols that we need to follow, and there's contract manufacturers that do this work. So it's not, it's an untrodden path for us as academic researchers, but it's pretty well established, and there's a lot of expertise in the Galway area as well. And your question about biofouling, similar to the formation of the fibrous capsule, you know, our idea is that we won't get this buildup of stuff on the device if we are actuating it, and it will get kind of cleared with minimal inflammation. That's kind of our idea.
All right, are there any other questions in the room? All right, let's say thank you one more time to Dr. Dolan.
Thank you so much.